Good morning, good afternoon, and good evening to everyone joining us from different parts of the world. My name is Dr. Sundas Qureshi, and I'm delighted to talk with you about a topic that is central to understanding how cells respond to stress and maintain balance: oxidative load and the antioxidant defenses. Today, we'll explore the integrated approach of measuring total oxidant status and total antioxidant status to map the redox biology. Then we'll also try to understand the disease mechanisms and apply these insights in research models ranging from inflammation to metabolic dysfunction. If you have any questions during the webinar, please feel free to ask them in Q&A section. My team and I will answer them one by one as we go, and we'll also address any remaining questions at the end of this session. For any technical questions after the webinar, you can also reach out to me directly or to Elabscience tech support team at the email IDs which are listed here on this slide. And all of us are always welcome, and we are happy to support your work. Before we dive in today's topic, I'd like to briefly introduce the Elabscience company, which was founded in 2009 and is now a global life science company with a presence in over 150 countries. Multiple international certifications have been obtained, including ISO 9001, ISO 13485, and CE certification. And we have also obtained tens and thousands of product citations in internationally recognized peer-reviewed journals, including the Nature Group, Cell and Science. Since the launching of our first bio research reagents and ELISA kits in 2011, we have continuously expanded our portfolio like cell culture solutions and clear kits in 2013, then antibodies, proteins, immunohistochemistry and Western blot kits, metabolism assay kits, food safety and animal disease kits, and our strategy product, which is flow cytometric antibodies, over the following years. More recently, we have introduced some advanced tools such as induced differentiation media, exosome isolation kits, ELISpot, CellaQuant, and primary cell isolation kits, while also earning recognitions like CiteAb Supplier to Watch and the Kotler Award for the customer value. This timeline reflects our ongoing commitment to innovation and high-quality tools supporting the cutting-edge research, including the studies on redox biology, which we will focus on today. Now, here is the one of the quick announcement. All the attendees will receive a digital participation certificate as a record and of their involvement. And each attendee is also eligible for a free 48 test sample kit with certain conditions applied. For anyone who couldn't join live or wants to revisit the content, the full recording will be sent to all the registrants. Now, let me give you a quick roadmap of today's session. We'll start with where we'll cover the basic oxidative stress and redox biology. Then we'll see where total oxidant status and total antioxidant status measurements fit in real research. Finally, we'll go through the practical workflow of these assays, from preparing the samples to interpreting the results. By the end of this session, you should have a clear understanding of both the science and the practical side of these assays. Now, moving on to the first section, which is foundation of oxidative stress and redox biology. This section basically will lay the groundwork for understanding the redox biology. We'll talk about what oxidative stress is, why it happens, and how a cell balances the oxidants and antioxidants. And by the end of this section, most probably, you will understand the basic concepts. Now, the most important question is why oxidative stress matters. But before that, we should know what happens when this oxidative stress occurs. So oxidative stress happens when the amount of reactive oxygen species surpasses the cell's antioxidant defenses, upsetting the balance that normally maintains the redox homeostasis. When this balance is disturbed, cellular components such as proteins, lipids, and DNA can be damaged, which affects the normal cell function. This imbalance doesn't just impact the individual cell, it can also contribute to a broader tissue damage and is also linked to inflammation, metabolic dysfunction, and aging. So, over time, the persistent oxidative stress can drive the progression of many diseases by interfering in the normal biochemical and physiological processes that keeps the cells healthy. So keeping this balance in check is essential for maintaining the cellular stability and overall physiological functions of the cell. Now, the reactive oxygen species or ROS, these are highly reactive molecules which contain oxygen. Endogenous sources include the mitochondria and enzymes like NADPH oxidases, while the exogenous stressors include UV, radiation, environmental toxins, heavy metals, viruses, and even the processed food. At moderate levels, ROS participate in normal cellular processes. When their production exceeds the cell's antioxidant defenses, ROS accumulation causes the damage to lipid proteins and DNA, potentially triggering the cell death pathways such as apoptosis or necrosis. So the indicators of elevated ROS may include the oxidative modification to the cellular molecules, lipid per oxidation, as well as the protein damage. Other than that, DNA breaks and the overall signs of cellular oxidative stress can also be the part of it. Dietary antioxidants such as vitamins, flavonoids, and other compounds can also help neutralize these ROS and protect the cells from the damage. Now, cells do defend against reactive oxygen species on their own through a combination of enzymatic and non-enzymatic antioxidant systems. Enzymatic defenses include the SOD, which converts a superoxide into hydrogen peroxide. Then we have the CAT, which break down the hydrogen peroxide into water and oxygen. And then we have GPX4, which reduces the peroxide using glutathione. Non-enzymatic scavengers such as GSH, glutathione, vitamin C and E, and some other small metabolite complement these enzymes by directly neutralizing the ROS and supporting the enzymatic activity. Together, these system maintains the redox balance, preventing the oxidative damage to proteins, lipids, and DNA, and ensuring the normal cellular function. Redox hemostasis that represents the dynamic equilibrium between the oxidant load and the antioxidant defenses. When this balance is maintained, the cellular processes function normally and oxidation damage is minimized. But if the reactive oxygen species accumulate beyond what the antioxidants can handle, this balance ultimately tips, resulting in the oxidative stress, which ultimately disrupt the proteins, lipids, and DNA and impair the cellular function, contributing to disease mechanisms later on. The total oxidant status. Now the question is, what is TOS or the total oxidant status? This is actually the one which measure all the oxidants present in a sample as a single integrated value. Oxidants might include a wide range of species from radicals like superoxide and peroxide radicals to the molecules such as hydrogen peroxide, nitric acid, and some other acids. These oxidants can be generated by activated white blood cells, through the enzymes like NADPH oxidase and some other enzymes using the substrate such as NADPH, arginine, and some metalines. TOS actually captures the cumulative oxidative load from all these species, providing a broader view of oxidative pressure on the cells and tissues than looking at the individual ROS alone. The total antioxidant status, on the other hand, measures the combined capacity of all the antioxidants which are present in the sample. This might also include the enzymatic defenses such as superoxide dismutase, sorry. Then the catalase and glutathione peroxide, and some non-antioxidant like glutathione, vitamin C and E, and some other metabolites. So TAS reflects the overall buffering ability of the cells or tissues against the oxidative damage, providing a holistic view of how well the system can neutralize the ROS. And then there are some dietary antioxidants which comes from fruits, vegetables, and other foods further contribute to this capacity, supporting our body's natural defenses. So measuring the TAS along with TOS allows us to evaluate the balance between oxidative load and antioxidant protection in a meaningful way. Why we have to go beyond a single oxidative marker? Because measuring only a single reactive oxygen species gives a limited view of oxidative stress. So cells are exposed to a variety of oxidants, and looking at just one can miss the bigger picture. So holistic matrix like TOS and TAS captures the cumulative oxidative load and overall antioxidant capacity, providing a more complete understanding of redox balance. This integrated approach helps link the oxidative stress to multiple disease processes from neurodegeneration and infertility to the cardiovascular and metabolic disorders, while also highlighting the protective role of the antioxidants. Redox behavior varies depending upon whether we study cells, tissues, or the whole animal models. Each system reveals a different layer of oxidative and antioxidant regulation. In cell-based experiments, we can control the conditions lightly and see the direct effects on signaling and metabolic pathways. Tissue studies add the complexity, showing how the neighboring cells influence the redox balance. Animal models demonstrate the full physiological picture where the systematic functions like inflammation, diet, and some other factors like hormonal signals shape the oxidative stress. Here in this Venn diagram, it highlights where these insights overlap. So some observations are unique to a specific model, while others such as ROS buildup, antioxidant depletion, or impaired variability appear across all the systems. This aims to link these redox shift with a meaningful functional outcome and help guide which model is more appropriate for answering a particular research question. TOS and TAS together tells us how a system is coping, not just what is happening at a molecular level. When the oxidant pressure rises faster than the antioxidant defenses, cells shift from adaptation to vulnerability. That transition point helps explain why some models show resilience while others progress towards dysfunction. Using this balance, we can interpret whether the change is phenotype and it is driven by the excessive oxidative load, inadequate antioxidant response, or both. It also clarifies why similar stimuli produce different outcomes across the models. And by reading the TOS and TS as a coordinated profile, we can get a clearer view of where the system is sitting along the path from normal physiology to the stress, compensation, and eventually the disease progression. Now, in this second section, we'll explore how TOS and TAS assays are applied in the actual research. We'll cover some examples like inflammation, liver injury, and metabolic diseases, and see why integrated redox measurements provide a clearer picture than the single markers. Now, the oxidative balance is at the root of many physiological processes. As we just studied, that when the reactive oxygen species accumulate beyond the cell's antioxidant defenses, multiple systems are affected simultaneously. So, in the immune system, this drives an exaggerated cytokine response, fueling the inflammation. In liver, redox disruption sensitizes the cells to injury, contributing to hepatotoxicity. Metabolic pathways are also impacted, leading to energy imbalance and dysfunction, as seen in conditions like obesity and diabetes. Finally, the signaling networks that rely on the redox sensitive pathways can also be distorted, affecting the cell communication and function. These effects show why the understanding of oxidative load is essential in the interpreting of disease mechanism and identifying the points where interventions can restore this balance. Researchers often choose TOS and TOS because they provide a global view of oxidative balance rather than focusing on a single reactive species. This holistic assessment helps us understand how tissues and cells are coping with the oxidative pressure in a more meaningful way. So the TES or TOS measurements are not just practical and versatile, they are also compatible with multiple sample types and relatively straightforward to perform. They also allow the comparisons across different models or the treatment conditions, helping in linking the oxidative damage to the functional outcomes. So by integrating both the oxidant and antioxidant data, we reduce the uncertainty and limitations that come with relying on the single marker. This approach ultimately provides stronger and more reliable insights into redox biology and its role in the health and diseases. Okay, during the immune response, neutrophils and macrophages work together to eliminate the pathogens. Tissue resident M2 macrophages. These release the cytokines like tumor necrosis factor alpha and CXCL1, which also recruits the neutrophils to the site of infection. The neutrophils then produce the reactive oxygen species, superoxide, hydrogen peroxide, and some other acids to destroy the pathogens and amplify the inflammatory signaling. Monocytes that enter the tissue, they differentiate into M1 macrophages, producing ROS and cytokines to sustain the response. As the inflammation revolves, M1 macrophages induce apoptosis of neutrophils and shift back toward an M2 phenotype and restore the tissue homeostasis. This coordinated interplay illustrates how oxidative load together with the cytokine signaling drives inflammation and can be measured using TOS and TAS to understand the redox balance in the research models. In case of liver injury models, oxidative stress also plays a central role in driving the hepatotoxicity. Excessive reactive oxygen species can damage the hepatocytes, triggering the apoptosis and impairing the liver function. This redox imbalance then also promotes the inflammation and can contribute to fibrosis over time. By measuring both the total oxidant and antioxidant capacity statuses, we can get a comprehensive view of oxidative load in liver. These measurements ultimately help us link the redox changes to functional outcomes, enabling better understanding of disease mechanisms and evolution of the potential interventions. Now, in case of metabolic disease, oxidative stress is also a key driver. For example, in obesity, excess fat accumulation generates the reactive oxygen species that disrupts the cellular signaling. In case of diabetes, oxidative load contributes to insulin resistance and impaired glucose regulation. In case of non-alcoholic fatty liver disease, ROS promote the lipid accumulation in the hepatocytes, worsening the liver function. So by assessing the total oxidant and antioxidant here, we can capture the cumulative oxidative burden, linking the redox imbalance and metabolic dysfunctions and inflammation in liver diseases and for the obesity. As far as the drug screening and therapeutic evaluation is concerned, monitoring the oxidative stress is also crucial. Why? Because this helps us understand how the interventions impact the cellular redox balance. By measuring a TOS and TAS here, we can track the reduction in the total oxidant load and improvement in the antioxidant capacity, which ultimately allow us the comparison of different compounds and treatment conditions, showing which strategies effectively restore that redox balance. It also provides a clear quantitative readout to link the biochemical changes and the functional outcomes in cells, tissues, or the animal models. Here, you can see a conceptual example showing how TOS and TAS change over time under the stress conditions. For example, in the control group, both the oxidant and antioxidant levels remains relatively stable, reflecting the balanced redox homeostasis. Under the stressed conditions, total oxidant increase steadily while the antioxidant capacity decreases, indicating a shift toward the oxidative stress. So tracking these trends help us connect the redox imbalance with biological outcomes such as inflammation, tissue injury, metabolic disruption, and highlights the value of integrated TOS and TAS measurements for interpreting the experimental results. Okay, using a simple oxidative marker, because there are several single markers which are available in the market, they gives the limited insight into the cellular redox balance and can sometimes be misleading. So by integrating both the TOS and TS measurements, we can capture the load as well as the overall antioxidant capacity. This approach provides us with a clear mechanistic understanding, stronger connections to biological outcomes, and more reliable, relevant data sets. Essentially, integrated redox readouts allow us to make better evidence-based conclusions about the oxidative stress and ultimately its impact on cells, tissues, or disease models. Summarizingly, TOS and TS measurements provide meaningful advantages across diverse research areas. They also help in tracking of oxidative stress and inflammation, liver injury, metabolic diseases, and the therapeutic interventions, which ultimately offers us integrated insights that strengthen the conclusion and improve the biological relevance of the experimental studies. Okay, now in this final section, we'll focus on the practical side. I'll walk you step by step through how Elabscience TS and TOS kit work and how to prepare the samples, interpret the results, avoid some common mistakes, and ensure the data quality. First of all, why we assess the TES and TOS together and how how we can interpret the shifts. Measuring just the oxidants or antioxidant individually will limit the view of cellular redox state. So by combining these two in the same study, we can gain the complementary perspective, capturing both the oxidative load and antioxidant capacity. There are different patterns which can emerge. For example, an increase in the oxidative load with the simultaneous decrease in the antioxidant defense, or a mismatch ratio where one changes before the other. So observing these patterns together help us understand the real impact of oxidative stress on tissues and cells. This integrated approach provides the mechanistic insight and make our conclusion more biologically relevant and help us connect the redox changes to the functional outcomes. Now let's take a closer look at Elabscience TOS and TAS kits. The TOS kits measures the total oxidant load in the sample using a colorimetric readout at 590 nanometers, while the TS kit assess the total antioxidant capacity with the detection at 650 to 670 nanometer. Both the kits come in flexible formats ranging from 48 test kit to 500 SS, so you can choose the size that fits your study needs. These kits are also designed to give you a complete picture of the redox balance. Now, with this information, you can plan your experiments knowing the expected detection ranges, readout methods, and integrate the data effectively in your study of the oxidative stress and the cellular resilience. Now let's look how these assays actually work. So starting with the total oxidant status, the assay measures the overall oxidant in your sample, whether it's cells, tissue, serum, or some other fluids. Under the acidic conditions, oxidants in the sample convert the ferrous +2 ion to ferrous +3. This ferrous +3 then reacts with xelenol orange to form a blue-purple complex. The intensity of this complex or this color measured around 590 nanometers directly reflects the total oxidant load. On the other side, for the total antioxidant status, it quantifies the cumulative antioxidant capacity, and it will in this assay ABTS, which is first oxidized to a green radical cation, which is ABTS positive. Then the antioxidant in your sample reduce it back to the colorless form. You can measure the absorbance at around 660 nanometers here or to 670, and then we can determine overall antioxidant strength. There is another vitamin E, which is Trolox. It is used as a reference standard to give us a consistent comparison. Then these two assays, TOS and TS, they will provide us the complementary view of the redox balance finally. Now, this kit comes with all the components needed for a complete assay. For a TOS, you have a chromogenic agent, substrate, and hydrogen peroxide standard, along with a microplate and the sealing film. For TAS kit, it includes the buffer solution, chromogenic agent, Trolox standard, microplate, and the sealing film. Each kit is available in different sizes to suit your experimental needs. Like a 48 test kit, 96 test kit, and or the 500 essays. And this is the 98 test kit, which is being offered as a free sample for all the attendees who are attending our webinar. It is also very important to store the reagents according to recommended conditions. For example, for the TOS, most of the reagents are kept at 2 to 8 degrees and protected from light, while TS reagents must be stored at -20, also with the light protection where specified. So these specifications are mentioned in the manual, so you don't have to worry about anything. And it is always recommended to follow these guidelines to maintain the reagent stability and ensure the reliable assay performance. Okay, when using these assays, the workflow is straightforward and compatible with multiple sample types. The reaction is colorimetric, which means that the degree of the color change reflects the amount of oxidants or the antioxidants which are present in a sample. The TOS assay is measured at around 580 to 590 nanometer, while TAS can be measured at 650 to 670 nanometer on your microplate reader, reflecting their distinct reaction chemistries. Supported sample types might include serum, plasma, cell culture supernatant, and tissue homogenates, which allow the application across a wide range of biological systems. The general workflow is very easy. It typically involves the preparing of reagents, generating the standard curve, preparing your samples, adding the standards and samples to the plate, and finally measuring the optical density in your microplate reader. So if you follow these steps, it will ensure the accurate and reproducible results. Okay, when we analyze the TOS and TOS results, we rely on the standard curves to translate the absorbance readings into a meaningful concentration. For TOS, as I have just mentioned, that the sample is compared at the absorbance rate of 590 to calibrate the curve, which is generated from known hydrogen peroxide equivalents. For TAS, absorbance 660 to 670 nanometer, which is reference against Trollocs standard curve. For the liquid samples, the calculations are straightforward, taking the slope and intercept of the curve and applying any dilution factors. For the tissue or cellular samples, we have to normalize the values to protein content or the tissue weight. This is the key to ensure that your standard curves are properly fitted and that blanks are correctly subtracted. Smaller mistakes in the curve creation, dilution, or sample handling can significantly skew your results. So you better have to pay attention to all of the small mistakes. Okay, when we are running the TOS or TS assays, there are several common issues which can arise. First of all, pipetting errors, they can lead to usually high or low readings. So always ensure that the pipettes are calibrated and techniques which we are using are consistent. If the standard curve is not linear, it is usually indicating incorrect preparation of the standards. So prepare fresh standards and verify the concentrations every time. Then a high variability between the replicates often comes from inconsistent incubation times or the sample handling. So make sure that all the samples are treated equally and mixed properly. For example, in TAS assays, low signals may result from interfering substances in the sample. So consider diluting or checking the compatibility. Then on the edge wells, sometimes differ from the central wells due to evaporation or handling. So always use a sealing film. It will minimize the delays before the reading can help. Then finally, we have to slow or fast color development. This usually points to reagent temperature issues or the expired reagents. So allow them to equilibrate and always check the expiration dates. By anticipating these issues and following these solutions, you can maintain high quality, reproducible results. These colorimetric assays offer several practical advantages for the researchers. First of all, they are fast and the results can be obtained within a short incubation period, allowing the efficient throughput. Secondly, they are robust and reliable. The color change provides a reproducible readout that minimizes any kind of variability. Finally, these assays are accessible for nearly every lab setup because all the labs must have a microplate reader without the need of any specialized equipment. So this combination of speed, reliability, and accessibility makes the colorimetric approach an ideal for the routine TOS and TS measurements in the diverse research applications. Now, to wrap up, remember that oxidative stress occurs when the reactive oxygen species outweigh the cellular antioxidant defenses, disrupting the redox balance and impacting health. Measuring TOS and TAS together gives a full picture of this balance, offering stronger insights than the single markers alone. Then the integrated redox readouts also help us draw more mechanistic conclusions and better understand the disease models or the interventions. So by following these practical workflows and using the reliable assays like Elabscience TOS and TAS kits, researchers can generate a reproducible high quality data. With also, we can keep these principles in mind when planning the studies and use the TOS and TAS measurements to strengthen your conclusions about the oxidative stress and the cellular resilience. That's all for today, and thank you very much for joining us from different parts of the world. Here are some of more information about Elabscience, why you should choose it, and we have mentioned all the details about address and our social media here, so you can contact us through any channel. Now, as far as the questions are concerned, we are open for questions now. So if you have any questions, let's talk about one by one. So here we have one question. Can TOS and TAS be used to compare the oxidative stress levels across different experimental models or species? So I have mentioned this in my presentation as well. And yes, because the TOS and TS reflects a global oxidant and antioxidant status, they are well studied for the comparative studies across cell lines, tissues, animal models, and even the clinical samples, provided the sample preparation and normalization strategies are consistent. So, okay, here we have another question. How sensitive are TOS and TAS assays to small biological changes, such as early stage stress or mild interventions? Okay, for this kind of question, it is important to know that these assays are sensitive enough to detect any subtle changes in the or subtle shifts in the redox balance, especially when the changes in the oxidant load and antioxidant capacity occur together. This makes them useful for the early stage stress models and also for the monitoring of the gradual response to the treatments. Okay, now we have another question here. When designing a study, at which stage some of the at which point the TOS and TS measurements are most informative? Actually, the TOS and TS are particularly informative when measured at the baseline and at the multiple follow-up time points. This approach captures the redox dynamics over time and help distinguish the transient oxidative responses from the sustained redox imbalance which occur over time. So I hope that answers your question. So do we have any more questions? We'll wait for more questions for a little while. Okay. Okay, I think there are no more questions. So thank you very much, everyone. And you will get the recording, all of those who have registered for this training. And if and all the attendees, you will be getting a free sample. I will send the emails stating all the conditions. So you and the form, and then you can send us your requirement, and we will send you the free sample. And also I'm going to send the participation certificates for all the attendees. And other than that, I just want to inform you that all of these could take around 1 to 2 weeks after the training. So just stay tuned, and we will get back to you very, very soon. Okay, thank you so much, everyone. And goodbye. See you next time.